Assalamualaikum warahmatullahi wabarakatuh. Good morning, Professor Dr. Mukteningsi, as the lecturer um, of the subject bioscience. In this occasion, we are from the 9th group are going to present our presentation about Animal Genome Project, and we're going to provide our arguments in opposition or the contradictions about Animal Genome Project. Before we start, here are the members of the team. The first one is Fuad, the second one is Mulia, and the third one is I am Shyla. Here are the outlines of the arguments that we are going to present in opposition to Animal Genome Project. The four first arguments are going to be presented by me, and the others will be presented by the others. The first one is about ethical concerns. The second one is genetic discrimination, and the third one is misuse of information, and the fourth one is environmental impacts. We start from ethical concerns. Is it ethical to create diseased animals that are very likely to suffer? Diseased animals are the results of the project. Is it ethical to create them that are very likely to suffer because the project might have some negative consequences that we might not know that we might not know yet. Okay, before we start to animal welfare, the Animal Genome Project is a scientific effort aimed at decoding the entire genetic makeup of animals. And this project has the potential to revolutionize our understanding of animal genetics and could lead to significant advancements in fields such as medicine, agriculture, and conservation like taman safari and etc. And however, despite its potential benefits, there are several contradictions that arise from this project, such as the main one is ethical dilemma. Is it ethical to create them that are fairly likely going to suffer? And the Animal Project requires thousands of animals as their subjects to be used in the experiments, which raises questions about animal welfare and the ethics of using animals for human gain. Animal welfare. What is animal welfare? Animal welfare based on the science of well-being and productivity of an animal. This involves the humane treatment of animals. So animals should be treated humanely, just like how human is treated. And scientists involve manipulating animals for human ends as if the animals were nothing more than human property rather than treating the animals as being the value in themselves. So the animal welfare means that experiments that are not treating the animals for the project or for every experiment that treats animals not humanely should not be conducted, because it is contradiction to the animal welfare. And recent action to allow animals to be patented reinforces the idea of animals as human property rather than beings in their own right. While the project is aimed at improving animal health and welfare, the methods itself used to achieve this goal may have unintended negative consequences for the animals involved in the experiments. This contradiction highlights the need for ethical consideration in scientific research, especially um, when the animals are involved in the experiments. We don't know if the animals are going to suffer or not, or the bad or the negative consequences that will happen to the animals that will waste its life. Because animals, after all, are the same like us. They breathe, they are alive, and they deserve to be treated as we are treated, as we are being treated, but humanely. And then another contradiction is the potential for genetic discrimination. The decoding of animal genomes could lead to the identification of genetic variation that may cause certain diseases or conditions. And then these informations could be used to discriminate against animals with certain genetic traits. Like in the picture, every dog can be a good dog. And because these days people are biased by some breed that are maybe some breeds that are maybe viral, and then um, they have the kin to have the breed and etc. And that could be included as discrimination. So this could be this could int this could ultimately result in the loss of genetic diversity in animal population that could have negative consequences for the health and survival of these populations. And then this contradiction highlights the need for re re responsible use of genetic information. Genetic discrimination also leads to a misuse of genetic information, where inappropriate use of genomic data poses very specific risks, since it can be used to identify an individual of animals. And genetic information can be also used unfairly to discriminate against or to make the stigma of animals. The next one is a concern about the potential environmental impact. There are two that I and the team think or argue that there are two during experiment impacts and the post-experiment impacts. The during experiment has impacts to the environment because the production of genetic data requires large amounts of electricity, and the disposal of waste products from the project could have negative consequences for the environment. Meanwhile, post-experiments where let's say that the project results an animal like a dog alive, a dog that is alive after the gene was modified. The genes were modified. and the dog itself might be danger to the dog itself in for having maybe several diseases that we don't know yet, or the dog can be a danger to the surroundings. There are 2 points for the post experiment: transfer of genetic material to wild populations, leading to a loss of biodiversity, because these modified genes uh, are going to meet the other ones that are not, that the genes are not modified, and then they will mate, and then they will have a pregnancies, and then the kids of the children of the animals will be different ones from the native animals of the wild, of the habitat. And then the second point is diffusion of toxic effects on other organisms. And after all environmental impacts, this contradiction highlights the need for sustainable research practices and the importance of considering the environmental impact of scientific research.
The next topic is new disease, banning of improved GM varieties, then no approval from the consumption. The eighth topic is incomplete experiment. The ninth topic is xenotransplantation rejection. The 10th topic is genomic gap. 11th topic is genome information. And the last topic is high research cost. Okay, so the next topic is new disease. It is undeniable that this project will create a new problem in the world that might arise from BSA, SARS, and new strain of avian flu, which is the fear might lead to a human influenza pandemic. The next topic is banning improved GM varieties. Today, genetically modified products are routinely used in many food products in US without labeling or restriction. However, in Europe, their use is being challenged. This situation has transferred to developing countries where, despite the advance brought about by the Green Revolution, there are individuals and countries who are now advocating banning the better GM varieties for use in Africa. The next argument is no approval from the consumption. Already a transgenic Python pig with expressed Python in its saliva and hence produce less phosphor in manure has been developed in Canada. Although this pig model is excellent, no approval from the consumption of such pigs is allowed to that in Canada, where it was developed, or in the US. Okay, the next argument is incomplete experiment, where in developing countries, the use of genomic will be implemented more slowly as production systems are modified to be more capitalized. Advanced genetic stock from developed countries will be used more frequently, and increased production can be expected from the genomic lead improvement. Traditionally, such actions have not always been successful because Western breeds are not well adapted to the environment of developing countries, which have different feedstuff, weather, and disease condition. The next argument is xenotransplantation rejection. Shortage of human tissues and organ available for transplantation has fostered an interest in xenotransplantation, and the pig is preferred donor owing to its size and comparative psychology. Recent concern about retroviruses and difficulties in producing transgenic pig meeting the standard required for safe transplantation has slowed the progress in the use of the pig for xenotransplantation and have caused some companies to scale back active research in this area. And the recipient the recipient mount a powerful rejection response to such foreign organs.
High research costs. The high cost of the research is one of the shortcomings mentioned by the community. For example, in 2018, to obtain genotype data from bovine DNA consisting of 15 variants, it cost about 116 US dollar. Genomic data analysis. Genomic data analysis requires genomic information from many livestock, so that analysis research have a high level of significance based on the research on the Frisian Holstein cattle population or the minimum number of cattle sample for analysis genome structure. Analysis require a minimum of 75 head in order to get analysis research with high accuracy. However, with the high cost of the research, this is a problem.